Hello, everyone. Like she said, my name is Mathew Fischbach. I'm a PhD candidate in Dr. Chad Meyer's lab here at U of M. And today I'm going to be showing you work that I've done as a visiting graduate scholar at the Mayo Clinic in Rochester, Minnesota, in the lab of Dr. Laura Rogers. In this work, NetCIS is a network-based common insertion site analysis for case control Sleeping Beauty for genetic screens. So 4 genetic screens are unbiased screens that use this observable phenotype of interest and work backwards to discover the underlying genes that causes that phenotype of interest. And Sleeping Beauty is just one way that a 4 genetic screen can be done. Sleeping Beauty is a 2-component system that can induce both gain and loss of gene function. The first part, shown here, is the transposon. It's a DNA cargo that contains an overactive promoter site for overexpression of genes, as well as a polyadenylated tail for gene loss of function. The second part that isn't pictured is the transposase enzyme that excises the transposon and inserts it randomly into TA dinucleotide sites within the entire genome. Now, the gain of function can happen in 2 different ways, as long as the transposon is inserted within the same direction as the gene. First is the full transcript overexpression by inserting before the first exon. And the second way would be overexpression of a truncated gene product by inserting between exons. And the loss of function can happen in either direction that the transposon is inserted in, as there are 2 splice acceptors that can lead to the disruption of the gene product. And once inserted, the transposons can be sequenced via the inverted repeated regions on the left and on the right, denoted as the IRL and IRR respectively. Then some bioinformatic analysis is needed to determine where the insertions are and their importance. Now, we've begun doing screens that utilizes both cases and controls. And what we want to see is how do they differ from each other. For example, we could be comparing Sleeping Beauty insertions found in mutagenized T cells within solid tumors versus T cells in the spleen that were not able to travel into solid tumors. And in order to analyze this data, there are a variety of bioinformatic tools out there for Sleeping Beauty analysis, such as Transit, Tratis, Tapdance, KCRBM, and IAS. However, each one of these comes with specific problems, and none of these tools on the left is without one of these following problems. The first is that insertion sites must be annotated before any statistical analysis, and that also requires that reads have to map to a reference TA site, or they are excluded from the analysis. And then some tools also require that if a read can't be mapped to a gene, then they will just throw those reads out as well. Whereas in NetCIS, what we are doing is that we're annotating our insertion sites after statistical analysis as we try to minimize the impact of annotation quality. And we allow insertion sites to annotate to either multiple genes or even no genes, and we don't throw out any reads or insertion sites that can't be mapped to a reference TA site. The second is that statistical tests may not reflect actual data, as a lot of these tools would assume some sort of statistical distribution that doesn't actually reflect the underlying data. Whereas with our case control experimental design setup in NetCIS, we'll be able to just compare the distributions between cases and controls easily. And lastly, a lot of these tools are inefficient, outdated, or unmaintained, while NetCIS is, well, now brand new. It's efficient, updated, written in Python, and once the reads are mapped, it can just run in minutes. So the NetCIS pipeline begins with the FASTQ sequencing files and then pre-processing them, trimming off adapters, mapping the reads, filtering and quality control, as well as normalizing the reads to count per million. And then we generate our PSIS networks, or our pseudo-common insertion sites. And these are regions of insertions that are grouped together based on their proximity to each other, based on some sort of genomic distance. And we call them pseudo because we've not yet evaluated them for any statistical significance. And it's at this point that we keep the case and controls reads separated and process them separately into their own PSISs. And we do this by creating an undirected graph of insertion sites per chromosome for the case and control, and identify individual subgraphs as our pseudo-common insertion site. Then we go on to discover the common insertion sites, which are typically sets of insertions that are expected to have the same sort of functional impact. And in this case, a common insertion site, a CIS, will be where a case PSIS overlaps with the control PSIS, resulting in a set of insertions from both cases and controls that we will use in that common insertion site. So it provides us with reads that we can compare with the Wilcoxon rank sum test to see if the two distributions are drawn from the same distribution. And we also look at the Fisher's exact test to see if there's an association between the occurrence of a CIS and the case control statuses of samples in that CIS. Then we can analyze our common insertion sites, annotating genes to a CIS, volcano plots for the statistical tests, ranking them, and having a candidate gene list outputted that then users and investigators can investigate further with those results. And this was just a very broad overview of how a PCIS is generated. So let's get a little bit more into the details of that process. The first part is that each insertion site is added as a node to the graph with additional information such as the total read count at each insertion site. After all nodes have been added, the genomic distance between each node and all other nodes is calculated, and edges are added between the pairs of nodes with a distance below a given threshold. And in this little example case, that would be a threshold of 10 base pair. The connectivity between nodes is then used to find the individual subgraphs within the larger chromosomal graph. And each of these subgraphs would then be called our PCIS and includes the set of insertion sites that spans a certain genomic distance for each one. So in this case, from our 8 insertion sites, or 8 nodes, we would have these 3 blue, orange, and green colored individual pseudo-common insertion sites. A common insertion site, a CIS, is found by testing for any overlap between the genomic regions of a case PCIS with a control PCIS. If an overlap is found, then each set of insertion sites within that PCIS is expanded as the union of the insertion sites between the two. And a read count of 0 is then recorded for insertion site that was observed only in the other group, as seen by these open circles between the two here. So this enables modeling of insertion sites present in one PCIS but not observed in the other. This also yields a defined genomic region as our CIS, where the number of insertions in cases and controls can then be tested for statistical significance. And even if a PCIS does not overlap with another PCIS, we can still test for significance as we've created the set of corresponding 0 read count insertion sites. In creating NetCIS, we needed a case control Sleeping Beauty screen to build NetCIS with. So we sought to reanalyze Rogers's 2020 Sleeping Beauty screen that focused on identifying common insertion sites that enabled T cells to better traffic into solid tumors. So they first set this up by crossing CD4 Cree mice with Sleeping Beauty mice to only allow transposition in CD4 positive and CD8 positive T cells. Then they injected bilateral tumor cell lines and harvested bulk tissue consisting of both left and right tumors, as well as the spleen, and sequenced them. The idea then is to find common insertion sites that differ between the tumors and the spleens, where the T cells in the tumors are the cases, and for the spleen, which holds a lot of the circulating T cells, would be our control. And an analysis of these insertion sites resulted in gene candidates, in which 3 genes, AAK1, SBRR1B, and EHHADH, were subsequently biologically validated as increasing the number of CD8 positive and CD4 positive T cells within solid tumors. Now, this is just great for us since we can use these validated genes as our gold standard. If we can find that these genes are ranking highly, we can kind of be confident that NetCIS is working. And to do this, we ranked all of our common insertion sites using a rank aggregation of 3 metrics: the rank sum p-value, total read counts of a common insertion site, and the total number of samples in the common insertion site. As we hypothesized that these metrics would reflect important and interesting common insertion sites that we would want to look into. And this method did prove fruitful, as the 3 validated genes, AAK1, EHHADH, and SBRR1B, were found to be in the top 15 of our aggregated ranked common insertion sites at 2nd, 6th, and 13th, respectively. So this to us confirms that NetCIS is working properly. So now we wanted to look at the common insertion sites that were found to be statistically significant from the volcano plots. And these show the resulting FDR values for every common insertion site using the rank sum test here on the left and the Fisher's exact on the right. Those that have a higher read count in the spleen are colored in blue, and those with a higher read count in the left tumor are colored in yellow. And any common insertion site above the gray dotted line would have an FDR less than 0.05. So a pretty stringent cutoff that kind of gives us these sparse looking volcano plots. And in rank sum, what we find is that a MER101C, a non-coding microRNA that shows to have the largest log FDR out of all of our tested common insertion sites, We also find some genes like PTPRD, a protein tyrosine phosphatase receptor type D, which is just involved in various signaling pathways, as well as SPEAR4, which in mice is part of the spermatogenesis associated expressional related gene family, and INSEL6, which is insulin-like 6 in mice, part of that insulin relaxin superfamily of genes. For Fisher's exact test, we had a little bit less, but we did recapitulate PTPRD as well as the SPEAR4D gene, and then we had the SPEAR4E gene that also showed up. But what we really wanted to look at were common insertion sites that ranked highly in NetCIS but were not found from the previous 2020 analysis. So we chose 3 interesting looking common insertion sites to look at a little bit further by mapping the insertion sites onto just the genome here. And the first one that we looked at was our top #1 ranked common insertion site that was not annotated to a gene. And what we found was that this common insertion site was upstream of CREB5, which functions in both T cell development and migration. Now, we initially thought that CREB5 was going to be too distant from this common insertion site to be a functional target. However, the region of that common insertion site shares significant sequence identity with the first intron of the human CREB5 ortholog. So this suggests to us that there could be the presence of a conserved regulatory sequence that's annotated in the human reference genome, but is unannotated in the mouse reference genome. In figure B, we then looked at PTPRD as it was ranked 17th overall and was significant in both the rank sum and Fisher's exact tests. And the Sleeping Beauty mutations in PTPRD shown here appear to be disruption of expression events, such that the loss of PTPRD is hypothesized to improve T cell infiltration. And when we looked further in the literature, we found that PTPRD is a negative inhibitor of STAT3, which would, with the loss of PTPRD, would increase JAK-STAT signaling. And JAK-STAT signaling has been shown to improve the persistence of therapeutic engineered T cells, which then supports a potential role for PTPRD loss for intratumoral T cell accumulation. Lastly, with figure C, we chose the MER101C as this was highly significant in our rank sum test, was ranked 9th overall out of our common insertion sites. And while published literature suggests that MER101C could be biologically relevant to T cell function, more investigation is needed as this was found in a region that was known to be biased for mapping. So overall, what NetCIS accomplishes is that it directly compares cases to controls for insertion sites and avoids pitfalls of other tools. We provide separate statistical and ranking results, and we've able to recapitulate previously validated genes, and we are overall very efficient and can run quite fast when looking at thousands of common insertion sites. So with that, I'd just like to acknowledge my lab members from both the Myers lab and the Rogers lab, as well as the funding this project has received. And with that, I'd be happy to take any questions.
Seems like your choice of controls is key to how you're doing your statistical analysis. So could you talk a little bit more about how the controls are selected and potential sources of bias that might come from that?
Yeah, for this instant, the controls are just the T cells that are in the spleen. So they could have some T cells that would also have been able to traffic intratumorally, but the large majority would just be the circulating T cells within the body that have not been shown yet to be able to do that. So there would be some signal that could be possibly between the two. Misleading in ways, yeah. But with enough cases and enough controls of which, I didn't say, but we had, I think it was about at least 100 more for each. Yeah, that's true. Yeah, I could just lie, couldn't I? No, it was over 100 for the samples that we had. So it was, I feel like that's probably good enough to be able, and then we combined all of them together from all those samples into these common insertion sites. So we're taking from many samples. Yeah.
Thank you.
Thank you.